Bonjour, bienvenue. Thanks very much for joining us for another weekly research conference at the University of Ottawa Heart Institute and also throughout the university and in the region. And just before I start introducing our esteemed speaker, Dr. Morgan Fullerton, just a few logistics in that if you have questions, use the Q&A feature on the bottom of your screen. And you can do this throughout the presentation. And you can also ask your questions live at the end of the presentation as well. But please then use the hands up feature so that we can then invite you to join in by the moderator. And but if you have any technical issues, please use the chat function. And then Kelsey or Allison or others will be able to problem solve and troubleshoot for you to make sure you can join us successfully. We're delighted that Dr. Fullerton can provide with us in terms of a fantastic scientific update today. And he's a well-recognized expert, as many of you know, in lipid biology, with training in cellular, molecular, and biochemical techniques, focusing on cell metabolism. And he joined the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa Faculty of Medicine in 2014. And after completing his bachelor's studies in molecular biology and genetics at the University of Guelph, Dr. Fullerton transitioned to PhD studies under the supervision of Dr. Marika Bakovic. And using a novel knockout mouse model, his doctoral studies characterized the importance of phospholipid metabolism and its links to obesity and metabolic diseases. And Dr. Bakovic's lab has in-depth expertise in nutrigenomics and the choline transport in relation to phospholipid metabolism. And I think we'll hear some of the latest on that. And in the fall of 2009, he began his postdoctoral studies in Dr. Greg Steinberg at McMaster University. And Greg is really another cutting edge researcher, particularly focused on the understanding of the important protein named the AMP activated protein kinase, or AMPK, which particularly regulated obesity and metabolic stress. And many of you know the AMPK is the master regulator of intracellular metabolic programming, substrate utilization, and in turn, important processes of cell growth, adaptation to stress, autophagy, and cell polarization, and many other important functions. And Dr. Fullerton's laboratory is focused on uncovering the alterations in lipid metabolism in chronic diseases, such as diabetes and atherosclerotic heart disease. And specific areas include the role of, for example, major regulators such as AMPK in terms of macrophage cholesterol metabolism, and also choline uptake through agents such as CTL1 and their role in metabolic diseases. So we look forward today in the update on the topic of choline as a driver of macrophage immunometabolism. So welcome and thanks very much, Morgan, for presenting today.
Okay. Well, thank you very much. I really appreciate the offer, and this is, it's an honor to be following up on two. I've only caught the last two webinars for this series, but to follow in the footsteps of Dr. Rosani and Mulvihill, to colleagues and friends and just experts in their fields. It's a little bit humbling, if I'm going to be honest. And I'm also a little bit, I'm going to walk you through a few of my first slides, which are basically just going to reiterate what Dr. Liu has said. I kind of doing a little bit of an introduction, but hopefully it serves as a little bit of maybe just an informal introduction to the trainees in the audience, and sort of I'll to try to outline my path, which was shockingly not as direct as it could have been in terms of my willing it to happen. But to begin with, I would like to acknowledge and pay respect to the Algonquin people, who are traditional guardians of the land, and acknowledge that the university has a longstanding relationship with the territory, or they do, these peoples with it. We are on this land and it remains unceded. And I provided 2 headshots to Kelsey for this, and she chose the formal of the 2. But as most of you who will know me know, I don't take myself very seriously. And this was shot probably a month and a half, maybe 2 months into March, or when we started the pandemic. And I feel like it captures the essence of what we're all going through right now, where we're getting through. But without my 2 stuffies here, I'm not sure I would really have gotten through most of those times. So whatever you need to do, it's okay. And in the end, our perfect headshots aren't necessarily representative of how we're doing that day. So not to worry. And so if I'm going to just sort of walk you through and exactly what Dr. Liu mentioned. I did my undergraduate studies at Guelph, and to say that nothing happens during your undergraduate and master's and PhD isn't so true. I was able to meet my partner throughout, well, in undergraduate studies, and she stayed to do her master's and I stayed to do my PhD, but it all happened at Guelph. And so lots happened. We sort of laid the foundation for life throughout school, which is to say that that's a good thing. We can multitask by studying and also sort of getting through life. So then we transitioned, and my wife got a job in Toronto, which meant that the world that was our oyster for my postdoctoral studies became the GTA. And luckily, as Dr. Liu mentioned, Greg Steinberg was transitioning back from Australia, although it would have been nice to go to Australia to work with him. I went to Hamilton to work with him instead. And it really was just the most fortunate timing that could have ever happened, because I was able to sort of apply what I'd learned and learn infinitely more. And again, a timing with a lot of the projects just really meant that it worked for both of us. And so out of life in McMaster, it was really, we got a dog. And this was a big thing because I wasn't a dog person, but Gus is now the first member of our family. And so then I was very fortunate to actually sign on the dotted line in 2013 to start in 2014. And I actually remember the last time I gave a talk at the Heart Institute was November 30th of 2013. And I had a handlebar mustache that was very off-putting, I think, to some, but it was the end of November and I did it anyway, but there's infinitely more gray in the beard right now. And so in Ottawa, again, life continued. We were able to welcome not just one, but 2 new family members. And I always point to the dog's face. He was very happy about what might be the prospect of having this little companion. And now he's not so happy. He knows exactly his place has just sort of dropped another rung in the ladder. But in the end, life continues and we're excited for the next steps. And so in terms of the lab, we've actually gone through a little bit of we call it a rebranding. And in the end, we got so tired of saying Immunometabolism Lab that we put the 2 words together. We made a thing. It sort of stands for everything that we stand for, which is hopefully a lot of good things. And what I want to do today is to do, as Dr. Liu mentioned, is to sort of give an update. And it's nice to be able to reflect, to see sort of what we've done in the last few years. And it's a celebration, really, of all of the people who've been doing the work. And so I think it's really, for all the trainees who are now listening, generating questions is easy. You could sit and you could think of a question to anything, but following up on those questions is the hard part. So these are the individuals who have journeyed through the weeds and the trenches to try to get at the answers to the questions. And so, and some are playing sort of a leading role in this, and others like Julia and Tyler in this particular project have really been in a supporting role, but no less important. And so the lab, as Dr. Liu mentioned, we focus in on lipids because that's sort of my history, but we really transitioned into the link between immunity and metabolism. And I'll get into how those 2 things are linked and intricately related. But we sort of come up with these overlapping areas, and some of them are to do with, we're very focused on atherosclerosis and macrophages in particular, and their role in driving and potentially being a target for sort of alleviating some of the stresses in atherosclerotic heart disease. But it kind of overlaps in some of these metabolic regulators like AMPK and mTOR, and its control really over lipid metabolism in general, and in particular in the liver. And we have other projects in particular, but right now I would like to take you down this sort of roadmap of choline as a nutrient and its importance in different metabolic diseases. I think I amended my title a little bit because I have a little sort of vignette into something that's not, it's immunometabolism, but it's really more focused on lipid metabolism in the liver. If I focus in on choline now as something that I'll give a very brief overview, because it could be a snooze fest for some, but and we start off really with just some old white guys who were actually, it's amazing to me to think that in 1850 people could have been determining the chemical composition of anything just in terms of sort of how scattered my brain can get. But it was really in 1850 that some of these initial determinations of the composition of some of these things that we're now taking for granted in terms of phospholipids or lipid metabolism came to light. And so Theodore Goebli, Adolf Strecker, who, fun fact for all of you trivia aficionados, is that this individual also discovered for one of the first derivatives of metformin. And so they are intricately linked to the saga that is metformin. But if you fast forward, I think we're all familiar with Charles Best, and after working with Banting and the discovery of insulin, Dr. Best did a lot of the groundbreaking work on looking at choline deficiency and how that, or that it led to fatty liver disease. And this work was done in dogs. But for all of the importance that I can ascribe now to what choline does for the body, it really was only in 1998. I was in grade 12 in this, maybe even the grade 11, it was recognized as an essential nutrient. And I'll get into as to why the essential part comes in, but it really was, now that I think about the diversity that is not showing in terms of these three individuals, Dr. Stephen Zeisel was one of the big, I guess, drivers of choline and the understanding of the importance of choline and the person that he trained with. But I think now Mary Cottle, who's at Cornell, she's done some phenomenal work with stable isotope tracing and really understanding from choline after it gets into the body and looking at it in humans and some of these metabolic pathways from which choline is feeding into. So their work is really built onto the foundation that has been laid down. And so if we look from a general perspective, choline, I am not a nutritional scientist, but I think one thing that becomes very apparent is that if we look at across the generational spectrum, across the developmental spectrum, the necessity for choline as our body requires it, or the requirement for choline differs. And so from birth to 6 months, the majority of the choline is coming from the maternal diet or the supplemented diet that we're being provided. And now for 7 months, and that requirement is quite low. But as we age, the requirement sort of increases steadily until sort of early and late, early and then adulthood through. You can see that there's a sex-dependent and potentially a pregnancy and lactation-dependent requirement for choline, in that lactating mothers, pregnancy itself, you're not just supplementing or you're not requiring choline just for yourself, but for the developing fetus and for the newborn. And so if we look at choline, it's as a composition of food. I mean, this is something that I think companies can spin in different ways, but this is actually, I sort of fact checked this a little bit with some of the publications with there, and it's very accurate in that the majority of choline, either found in its phospholipid form or its free form, is found in animal foods. So eggs, meats, and it really is to a lesser extent in fruits and vegetables and different legumes, but which I guess people are advocating either in selling a nutrient supplement or from a health perspective, is that the estimation is that the majority of the population across different spectrums is actually deficient for their choline intake, which they use to sell their supplements, but also in certain situations, there may be some truth to the potential benefits for upping that intake to the adequate and required levels. But from our lab's perspective, not being a nutritional scientist and not being looking at it from a population level, we're really sort of focused in, almost laser focused on the effects and the metabolism of choline and how that's regulating and how it's regulated in different physiological contexts. And so just as a very broad overview, of which we'll get into a little bit through some of its downstream incorporations, it's important in various different neurological disorders, schizophrenia and Alzheimer's disease. Important in development through the kind of relationship with different neural tube defects, important in lactation in so much as it is required so much more during lactation. It feeds into folate and metabolism and into methylation, as I'll touch on. It's also used as a prognostic marker. So different isotopes that can be tagged in terms of radiolabels onto choline, because of, as I'll mention as I get into, choline serves as a precursor for different lipids that then drive the expansion of cells. It is like a prognostic marker for different cancers. And then I guess what we're more focused on, just from a sort of linear perspective, would be different metabolic conditions like diabetes and cardiovascular disease and fatty liver. But all of these have something in common, which is sort of that underlying immune component, which is what I like to focus in on today. So just again, to kind of finish up on the background in terms of choline metabolism, choline can get into the cell only through a transporter. So it requires active transport. And what we're looking at first here is the Kennedy pathway. So Eugene Kennedy, who trained with Krebs and did a lot of work past that, that's sort of owing, or we are owing a lot of what we know about phospholipid biosynthetic pathways to Eugene Kennedy and his team, in that when choline comes in, and the analogous pathways for another phospholipid, where ethanolamine comes in and follows, again, a mirror image pathway where it's phosphorylated, it's incorporated with CDP, it combines with diacylglycerol, which then ties it to other neutral lipids like triglycerides, and then it forms phosphatidylcholine at the end of that pathway. And phosphatidylcholine is the main phospholipid in all eukaryotic membranes, and without phospholipids, we just wouldn't be here. So the cells require choline to be able to then sustain their membrane. And when we look at the essentialness of choline in terms of a nutrient, the reason it's essential is that we can't make enough of it. And I say we can't make enough is because specifically only in the liver, this enzyme, phosphatidylethanolamine methyltransferase, is expressed. And so this enzyme methylates PE 3 times to form choline. And so by forming choline, or phosphatidylcholine from PE, we can then break it down to have choline as a newly derived synthesis. But we can't make enough of it in our liver to sustain the requirement sort of from a whole body perspective. So that's where the essentialness comes in. Choline can also be acetylated to acetylcholine, essential neurotransmitter. This is sort of what we're thinking is the main link to the neurological ties or the neurological disorders that I mentioned before. And in addition, what was thought to, and I will preface this as what was thought to only occur in the liver and kidney, is the oxidation of choline in the mitochondria. And once it's oxidized and then converted to betaine, this can be converted or combined with homocysteine to feed into make methionine, which then feeds into the methylation cycle. So choline plays a role in phospholipid biochemistry or biosynthesis. It plays a role in neurotransmitter biosynthesis, but also this sort of very known and established role in the methylation pathway, but again, linked to just the liver and the kidney. And I'll come back to that at the end. But one of the things that, as Dr. Liu mentioned, in my PhD, we looked at, before really my PhD, but my PhD lab was interested in how choline gets into the cell. And it can be transported by 3 family groups of proteins. And so first is this choline high affinity transporter, and this is almost exclusively expressed in neuronal tissue. And so it's transporting choline with unbelievably low affinities. And so it's a high affinity, and so you only need a little bit of choline for it to be recognized and transported in. Or it's transported in by these organic cation transporters, which are widely promiscuous. They just bring in anything and everything with the right charge. And so we're not really, as a cell, we can't really rely on this organic cation transporters. And so this choline transporter-like protein family, which is arguably not the best name for it, but these family members, and particularly CTL1, is one of the most highly characterized and recognized as one of the main choline transporters for phospholipid biosynthesis in a ubiquitous nature. And so just to touch on it briefly, we have tools in the lab that we're using to be able to test these processes. And so we have this thing, hemicalinium-3, which is a highly competitive analog of choline, and it blocks all of the high affinity choline transporters. So all of the choline transport like family and all of the high affinity through the CH2-1. But we also have a molecule that will inhibit the phosphorylation of choline, which is that first step along the Kennedy pathway. And so by doing this, we can really turn off the metabolism of choline as it feeds into the biosynthesis of phosphatidylcholine. And that allows us to really ask some questions scientifically in terms of what's the importance under various situations and contexts. And so again, as a quick vignette, I really just wanted to touch on two little stories before I get into the macrophage part, which is one of the first questions that we asked as a lab, and really it was driven by Connor and Rebecca, which was, what's the importance of choline, or what's driving hepatic choline uptake? And so we took from mice hepatocytes, we plated them, and for 24 hours, we sort of just stimulated them with various different metabolic stimulus in vitro. And then we did a very quick, just sort of a pulse of radiolabeled choline, and then we washed it off, and then we lysed the cells just to see, all right, under which condition is choline uptake different. We didn't want to look too far down. We did 10 minutes because after 10 minutes, we start to metabolize choline. Maybe it gets secreted by different lipids. And so other than the fact that we haven't really followed up on the insulin effect, although it makes sense that it goes up if insulin is stimulating lipogenesis in the liver, but under all these conditions, we saw that palmitate, the saturated fatty acid, really had a reasonable and very consistent and statistically significant effect at inhibiting choline uptake. So we really sort of went down that rabbit hole just out of curiosity, really, and that the presence of palmitate really was mirrored in the fact that in obesity, these saturated fatty acids are increased, and there's this sort of mimicking of the obese state in a dish. And so we could see that when we treated fatty acid treatment, which in this case we used a vehicle, we used an oleate and a palmitate combination, or just oleate by itself. If you look at the rate over time, really it was the palmitate that brought everything down. And that then was attributed, at least in vitro, to the fact that under the palmitate condition at the transcript level, we really saw that there was a feedback mechanism that was inhibiting the transcription of CTL1, which then fed through, and there was no sort of compensatory increase on the transcript at the translation level. And so we kind of attributed this decrease in the rate of choline uptake because there just wasn't as much transporter at the surface. And so there was a lot of sort of work around that, but in the end, we wanted to know, can we mirror that, or is it mirrored in the context of obesity in vitro or in vivo? So we took mice and we fed them a high-fat diet or the control low-fat diet for 10 weeks. So we took those mice and we sort of tried to, I guess, MacGyver up an in vivo choline uptake. So it's a hard thing to do with just a bolus of radioactivity, because that radioactivity can go anywhere. If we inject, which we did with this radiolabeled choline intravenously, it's going to then be taken up into various different tissues. It's going to get metabolized into various different ways. But by kind of doing a very similar thing to what we look at for glucose uptake in vivo, we were able to calculate that on a sort of micromole per milligram protein per minute, there was not as much choline that it was taking up and converted into hepatic phospholipid in the obese animal as compared to the lean. And so what we sort of confirmed was that there is this decrease in CTL1 protein, which again very well explains the decrease in the in the uptake. But interestingly enough, what we were able to see was that, independent of the fact that this is now looking at a control gene, we didn't see a difference in the transcript level. So there wasn't the same sort of continuity as there was in the cells. And so we reasoned that if we're seeing lower protein, but there's really no change in mRNA, then it must be a translational event. And so this is a controlled gene where we're looking at these different polysome fractions. Polysomes are the amount of ribosomes on a different transcript. And so we can fractionate them, and we've got kind of separated into these free ribosomes, or a monosome, free transcript, a monosome, which is really just one ribosome on a transcript. You kind of have these light or heavily translated areas that have a lot of ribosomes. And so this shows a really nice normal distribution, and really no difference between the two. What I don't have here is that the blue is the high fat diet. But if we look at the CTL1 gene, which is this SLC44A1, you can see that that's actually quite changed. And so we have this sort of skewing where there is less highly transcribed CTL1 transcript in the obese condition. And so there's some sort of translational feedback there. And so it's a published story, but I did want to give sort of a shout out to Rebecca and Connor, and Connor sort of drove the rest of that work. And really that stops the liver talk, and we're really sort of moving on and transitioning, I guess, to the next little vignette, which I've only ever heard people say vignette, and I actually don't know what it means, but I'm hoping that it means that it's a short story that is related to what we're wanting to talk about. To choline metabolism as it relates to hepatitis C virus infection. And so the short backstory here is that, and some of you will have heard this, and so I make the same joke, is that Kaylin, who was a very talented the first cohort of the TMM program back in 2017, he came into my office after doing a rotation project, and he says he wanted to do research on dengue fever, or even I think he said hemorrhagic fever. And at that point, I was ready to sort of kick him out of my office because I thought, no, thank you. But I took a minute and I thought, well, maybe if we can meet in the middle and there's a common ground, because we had sort of interests in the response of hepatocytes to different stresses that regulate choline metabolism, and choline metabolism feeds into phospholipid biosynthesis, and in the end that's going to cover a lot of different conditions. And so what we were able to do is to go down this sort of side path journey, invoking lots of favors and expertise along the way of different people. But to set the stage very quickly, hepatitis C, while there is now a legitimate cure for this virus, it has provided quite the health burden over time, and continues to in terms of accessibility and affordability of these antiviral drugs. And so hepatitis C is an instigator of chronic liver disease that can progress towards cirrhosis and potentially even liver cancer. And so there's still a lot to understand about the pathogenesis of this virus and how it interacts with the liver itself. And so it's that sort of viral liver interaction that sort of the bridge there, and the commonality for us was the potential for phospholipid to be that bridge, and for one to play off the other, or one to have a necessity of the other. So we asked a very simple question, and we're attending to ask very simple questions these days in terms of the reciprocal nature of these things. But does HCV infection alter choline uptake and incorporation into phospholipids? So it's very simply, yes, it does. And so we can see here over time that HCV infection in a model cell line, so these are human liver hepatocellular carcinoma cells that are immortalized. They're not the same as what we would find in vivo, but really the best that we have. And they've been altered a little bit to be able to be infected by these viral-like particles. And so we see that there's a decrease in choline uptake with HCV. So that's sort of one. And the uptake is different. We can see that lo and behold, and it really just sort of, we looked at the gambit, we looked across the spectrum of the pathways, and really just at the transcript level here for CTL1 was the only transcript that showed consistent reduction, but that it trends in the same direction, so that CTL1 expression is lower, accompanied with lower choline uptake. And that held true at the protein level as well. And so we again had this sort of situation where one was associated with the other. We can't really say that it was causal specifically, but certainly highly associated, that the lower the CTL1 transcript and protein, the lower we would also observe there being in terms of choline uptake. And so on the flip side, again, these are just sort of small points around the published story, but does limiting choline uptake and sort of blocking that input, does that affect how HCV replicates, or what's the pathogenicity of that virus? And so we took, again, in collaboration with lots of different experts across the country, just for sort of the small little paper, but we took HCV infected and we also inhibited with this hemicalinium, which was that sort of block of choline transport, and then looked at sort of an indicator of early viral replication. And again, just as a one-off, you can see that looking at HCV, you got sort of, you had the control, right? With no HCV, you get no viral replication. But and the control is here at 20 micromolar hemicolinium, which is our drug to block choline uptake. There's really no effect. But at the higher concentrations, which again, you never know what drugs are doing, but you see that it's actually augmenting the infectiousness of these particles. And so we were sort of faced with the bigger part of this story was that we sort of juxtapose these cell lines in the presence of fetal bovine serum, which is what we're all using for the most part, or human serum, which the group that we were collaborating with at the University of Alberta really done a lot of work to characterize that these cells, which are human in nature under human serum, behave quite differently. And so we were able to sort of, again, compare and contrast under these two conditions, what the necessity of choline and the effect of the virus on choline was for HCV. And so now we take a quick breather, because that was sort of the preamble of the work that I really just wanted to be able to champion on behalf of others, is now into the main sort of meat of what we've been doing in this project. And so it really, if you look at choline metabolism as this sort of nucleus, immunometabolism is the sort of the next step. And so this was work that was started by Shane, who was one of my first master's students, and he's now well on his way to being a resident after finishing med school. Caitlin finished the story, or at least helped finish the story, after doing a work study in co-op placement in my lab, and then went to John Pizzacchi's lab and did her master's and is now in law school. And this is my daughter. I don't even know at what age, maybe she's 18 months there, but she's now 7. And so we've been working on this for quite a while. And I just wanted to now sort of set the stage for as we go into this and talk about macrophages. Many of you are as excited about macrophages as I am, and whether they're not the most interesting cell in the world, we can debate. I'm not so sure myself, but they're certainly one of the most interesting. And in the end, they are not just monocytes, immune cells that differentiate into this sort of effector cells and respond to these danger and pattern associated molecular recognition or motifs that help recognize foreign antigens or even self. And these are so much more. And one of the things that I've always liked is to go back to the history of it. And so Eli Meshnikov, who, again, this should say 1916, but way back was sort of the first to describe, or at least the first that we know of, to describe that macrophages or some sort of cells could perform phagocytosis. And I urge the trainees in the audience to, for whatever they're looking at, to sort of delve into the history a little bit, especially if they're into macrophages, because there's a lot there that really, I think, would sort of heighten, or at least allow you to sort of hone in your appreciation for what you're doing potentially. But what we know now is a little bit more, it's not nuanced, it's complex, but we have a really good appreciation for the fact that macrophages, as they exist, are not just blood-borne immune cells that differentiate terminally once they get to an organ of interest. Macrophages are across all sorts of different tissues, and I would argue almost every tissue in the body. There's a resident pool of macrophages that have either been generated from the yolk sac or from the fetal liver or the bone marrow, that are playing essential roles at regulating homeostasis and keeping everything going, from the regeneration of senescent cells to infection and tissue injury. Which brings us to sort of a quick intro into what we know now as immunometabolism, which is the sort of intricate link between a cell's metabolism and an immune cell's function. And so I see this as two sides of a story, or a coin. And so we have metabolism that drives immune function. So this means that whatever the metabolism of the immune cell, whether or not it relies on its mitochondria to sort of pump out the ATP in a very efficient way and different substrates, or whether or not it relies mainly on the quick and efficient conversion of glucose to lactate to get a little bit of energy, but a lot of different biomass molecules to be able to just proliferate. Those metabolic programs can drive different immune pathways and effector functions. And that's very well established across almost every single immune cell. But the flip side to that would be the effect of that immune cell driving the metabolism of various different systems, various different organ systems. And so this is, I think, where sort of I distinguish it between cellular or systemic immunometabolism. And the systemic immunometabolism really does feed into whether or not there's adipose tissue dysfunction and liver, the effect of liver lipid metabolism or carbohydrate metabolism. And it feeds into all of the different organ systems. But for this purpose of this talk, we're really not going to get into the systemic aspect of it by and large, but from a cellular perspective, this is not meant to scare you, and I'm not going to go through it a lot, but there's a very good consensus in the field that macrophages exist across a spectrum. They are not just one or the other. There's no macrophage A, macrophage B. Macrophages are what we tell them to be, and in a lab, we tell them to be only responding to one or two stimuli, sometimes a cocktail. But in vitro or in vivo, they are seeing everything all the time, and it's constantly changing. And so it's a really hard thing to ascribe what we see in the lab to what we might see or mirror that in vivo, but we also have to start somewhere. And so one of the good conventions I think that's been adopted is being able to sort of tell the world what you've told your macrophages to respond to. And so in this case, we have a macrophage that's responding to IL-4 versus a macrophage responding to LPS and interferon, and two examples of what metabolism does or has been demonstrated to be at a certain time point. And my point to this slide is the fact that this is talking a lot about the different inputs of fatty acids going into the mitochondria. This is a lot about energy metabolism. But I would argue that metabolism is more than just energy. It's about what that energy is put towards. And in the context of lipid metabolism, this slide is meant to highlight the fact that there really isn't much known on the role of phospholipid metabolism or even the role of different immune stimuli on phospholipid metabolism in the context of regulating immune cell function. And so this is now kind of the narrative in my head is sitting back and going back to my very first publication as a pre-master student. I hopped on this project because it needed to be finished because Laura went off to med school and I was in the lab before the summer to start my master's. And so I really didn't know what I was doing. I was on this project, not really understanding that I knew THP1s were monocytes, but in the end, that they could have fed into a research program as I'm sitting here talking to you today was never on my radar. So it's maybe just, again, for the trainees in the audience, take a deep breath, and you never know what you're working on, how it could influence what you're going to do 20 years down the road. And so this paper, again, I look at it now and cringe a little bit because I would have done a lot of things differently, but two results I just wanted to tell you about because it really did set the stage for where this project started as I was starting in 2014, in that we showed that, so impaired trafficking, so we're saying that there isn't as much choline transporter like 1 protein, so CTL1 at the plasma membrane in THP1 monocyte-derived macrophages. We saw that as we differentiated THP1 cells, which are this monocytic human cell line, that when you stimulate them with different analogs of 4-bol esters or of diacylglycerol. So we hit them with a chemical that's going to then allow the suspension monocytes to adhere to the plate, and they adopt a lot of macrophage-like phenotypes. And so in doing that, in those cells adhering to the plates and becoming macrophages-like cells, we saw that choline uptake really goes down to the point that it was questioning of how are these cells doing this, and we showed that it was associated with disappearance of CTL1 from the plasma membrane. Again, I would have done things a lot differently knowing now what I know, but this result, when I revisited it, made no sense to me, none whatsoever. But I remember this being a very reproducible and robust response. But at the same time, I started to question it because I did want to question everything, and because in the end, when you activate macrophages, when cells go, I'm actually just going to scroll back up quickly to this slide. Monocytes are small. They're circular in circulation. They're not meant to be big and gangly like to be able to phagocytose everything, or they get stuck. Macrophages, upon differentiating, they expand. There's a lot of generation of membranes, and so in that that was the dichotomy that I couldn't get over. I couldn't get over the fact that monocytes to macrophages, as they differentiate, would need membrane of which choline provides the base component for. And in addition, it came out quite early, and I actually did this experiment when I was in master because I was just wondering what happens to fatty acid synthesis. Well, when we trace fatty acid synthesis, it actually goes up as we activate macrophages, which again, it's not differentiating them, but it's sort of that next step for convincing a macrophage to get a bit more reachy and to be able to phagocyte those things. And so this is a paper from Gerald Schmitz's lab where this is a monocyte and the membrane biosynthesis that happens. And they confirmed this finding as well, that fatty acid synthesis goes up as monocytes differentiate to macrophages. And so that didn't make any sense to me. And so I set out, as I started my own lab, the question that we really wanted to answer were, what is the sort of real, or under the circumstances, we're going to ask the question, what is the role of choline metabolism in macrophages? And we could really only ask it from two, again, reciprocal questions, and they're the same old questions that we ask, but we wanted to know how macrophage polarization affects choline metabolism. So the role of the external stimuli that immune cells see, how does that affect choline and its incorporation into its downstream targets? But also, if we limit the ability of choline to then be metabolized, does that have a consequence on macrophage inflammation? So I'm presenting this story kind of at the same time, even though we did it at the same time, but we've diverged a little bit. So in the end, the model system that we're looking at is bone marrow-derived macrophages. We hit them with a very large amount of lipopolysaccharide, which is a bacterial endotoxin, or a relatively large amount of the sort of anti-inflammatory cytokine, IL-4. And so again, we're telling them to be just one thing. They're not very, they don't have a choice, and it's not very representative of what they would be in vivo. But at the same time, we wanted to start somewhere. So this is just a very simple schematic, even though it might be busy. We were able to confirm that if we polarize macrophages with LPS, everything that we wanted to go up goes up. And if we polarize with IL-4, the macrophages that were polarized with LPS, they go in the opposite direction, but for the IL-4 stimulated genes, they go up. So nothing is going sideways as we're convincing our macrophages to adopt these sort of different spectrum phenotypes. But now we get into the really interesting questions, at least for me, is that what happens to choline metabolism? And so one of the things that I don't know if everybody thinks this way, but I had always come into this field appreciating that there were, again, these spectrums of pro-inflammatory or pro-inflammation, pro-resolving or anti-inflammatory, and that those things most of the time were opposite. If there was a pro-inflammatory response, the anti-inflammatory response needed to wait its turn before something needs to happen. So when it came time to stimulating with these sort of characterized pro versus anti-inflammatory cytokines or stimulus, I wasn't expecting the same thing to happen with both stimuli. But in turn, when we stimulated with LPS or IL-4, the rate of choline uptake increased under both conditions. When we looked at the kinetics, it was almost identical between the treatments in that they were upregulated. The affinity for choline didn't change, but the maximal transport definitely did. And there was no difference in the rate of PC degradation. And so that's just a trick that we used in terms of if there was a difference in the rate of degradation, then the end pool would potentially not be changed because it was being degraded faster. Maybe that's a mechanism to keep everything the same. And so that was just looking at sort of acute uptake, and we can see across the spectrum here over time, because we always like to incorporate time as much as we can. We saw that there was just more phospholipid being made in macrophages that were stimulated with either LPS or IL-4. And so as a quick interlude, maybe this was, again, just working on if phospholipid was being synthesized, then where are the fatty acids coming from? And the question of, is fatty acid synthesis augmented? And it turns out that yes, it is. We confirmed this again. I mean, it's been shown many, many times now. And in addition, what we're able to show is that there's actually quite a big skewing towards triglyceride, which again has been shown, but we were able to confirm it. And now, I'm going to take you into some lipidomics world that Dr. Bennett and her group helped us with, in the fact that I just want to draw your attention to the fact that we've now got macrophages that are polarized with nothing, polarized with IL-4 or LPS, and we did LPS and interferon as well, and that there's some differences, which is interesting and something to follow up on. But in the end, the differences are quite subtle, even in terms of what the different lipid is. And this is really to give out there an appreciation for the fact that you can have just a difference at a single bond and you have a different lipid. And so while we're looking at phospholipids in general, that the world of phospholipids is so intricately complex that it's almost it's very daunting to think about. But it's also exciting in that across these different polarization spectrums, there are some very stark differences that may have functional consequences that we might be able to follow up on. And so if we look just at PC, I mean, we see that with IL-4 and with LPS polarization, there's an increase in the total content of PC in particular. And what's kind of interesting is that the composition is actually very highly mirrored. I know this this interferon LPS plays a little bit of havoc in terms of comparison, but there are much more similarities between the composition of the IL-4 and LPS polarized macrophages than there are differences. And this is just a quantification of those things to see that they are actually the newly synthesized fatty acids that are the different ones. And so how is the choline entering the cell? Turns out that there is an upregulation of CTL1, but not CTL2. Again, CTL1 being sort of the focus of our investigations. And in the end, we were able to show that we upregulate choline metabolism or infection or models of infection. It upregulates PC synthesis, and that happens. But we wanted to know the flip side. And I might just skip through some of these because I do want to get to the end, and I always lose track of time when I'm monologuing. But if we inhibit hemicrylinium with hemicrylinium, where we have a primary antibody against CTL1, we can block choline uptake and PC synthesis. And what we could do is link that to diacylglycerol metabolism, in that when we inhibit PC synthesis, there's a redirection of diacylglycerol towards forming lipid droplets, which is quite interesting. And when we inhibit choline uptake with either hemicrylinium or CTL1 antibody, and we polarize with LPS in particular here, we can see we can augment TNF alpha and IL-6 secretion. And so there's this responsiveness. There's almost like this hyperpolarization towards secreting these pro-inflammatory mediators, where on the flip side, we're actually decreasing the pro-resolving IL-10 secretion if we do the same treatment. And what we could show is that if we treat with our choline blocker and we upregulate the amount of diacylglycerol that might be there and kind of shifting towards triglyceride, that is potentially activating, it does activate PKC, or at least there's higher PKC substrate motif recognition, and blocking hemicrylinium brings down, or at least attenuates partially, the effect of inhibiting choline on inflammation. So when push comes to shove, this is just sort of a schematic to show that we can sort of augment the feedback into NF-kappa B and pro-inflammatory responses by augmenting or by inhibiting choline uptake. And so the question is, what happens on the IL-4 side of things? And so does limiting choline uptake and metabolism affect macrophage IL-4 stimulated programs? And so we know that IL-4 upregulates choline uptake, but now what we wanted to know was what does a macrophage that gets stimulated with IL-4 really do? And what they do is, well, they stimulate and they express a lot of mannose receptor, arginase 1, this CHIL3, but in particular this retinol A or realm alpha. And it turns out that this was one of the most upregulated and most absolutely hammered to the point of almost no secretion gene that we looked at. And so we looked at mRNA transcripts. We could confirm that both hemicrylinium, which is our inhibitor, and you should never rely just on an inhibitor, but we were able to actually mirror that in a choline deficient media, this same realm alpha specific effect is present. And so we're able to attenuate and really just inhibit this IL-4 stimulated realm alpha response. And in addition to that, PD-L1 and PD-L2 are sort of markers that are sort of more upregulated in macrophages that are skewed towards being more pro or anti-inflammatory, depending if it's PD-L1 or PD-L2. And what we could see is that when we use our inhibitors, hemicolinium or RSM, we are shifting the spectrum a little bit. And so we're actually upregulating PD-L1 and downregulating PD-L2 with choline inhibition. And so now that was in vitro. And so what we wanted to know, and actually I'm going to show one more slide in vitro, which is from Maya, who did her undergrad, her 4th year project with us. And she actually went a bit further to characterize. We can actually say now that IL-6 should not be upregulated with IL-4 treatment at all. But in the presence of our RSM, so inhibiting choline uptake in the presence of IL-4, seems to dramatically upregulate IL-6. I have no idea why, but it's actually a pretty consistent result. And this new chemokine that we've looked at, CCL22, actually shows a very similar effect to realm alpha. So it seems to not just be a realm alpha specific effect, but it makes it no more clearer with these two involved. So very quickly, as I sum up here, we did an in vivo model. And so this was really taking advantage / benefiting from the collaboration from Dr. Mira Nair's lab at the UC Davis. And so they do an H. pylori, which is a worm or parasite infection that drives Th2 responses. It's an intestinal, in vivo and intestinal parasite that it's an in vivo model of an IL-4 stimulation. And so what they did was they infected the mice, but then did sort of a every 2 days injection of either a vehicle or choline uptake inhibitor for 5 separate injections. And then they monitored worm burden, and then on day 17, they sacked the mice. So there was a little bit of a weight difference. And then we actually let these mice go a little bit further and saw that these in a separate cohort, and there was a weight difference of RSM treatment, which is something that we're following up on independent of this. But kind of interestingly, from their perspective, is that there was no difference other than some sort of individual time points. There was no difference in worm burden and the number of eggs in the feces. And so that's an indication of the fact that by inhibiting choline metabolism, we're not really changing the ability of the infection to persist or to sort of be cleared more quickly or more slowly. So there's sort of a net neutral effect. But what we're able to see is that when we look at this choline inhibited group of mice and looking at their peritoneal cells, and no difference in the total, and this was repeated in 3 individual cohorts. And so this is just representative of 1 with an N of 5 in each, but we're seeing a dramatic an upregulation of markers that are found on macrophages that are tending to be more pro-inflammatory. So PD-L1 and CD86. But a downregulation of markers that are, again, more typical of macrophages that are more pro-resolving or anti-inflammatory, so PD-L2 and CD301B. And interestingly enough, if we look at the restructuring of the total immune landscape, I just wanted to draw your attention to these peritoneal macrophages. There's a complete shift between the vehicle treatment and the RSM treatment, that these populations, they go somewhere, and whether or not we are just not being able to assess them, to see them, or whether or not they are actually traveling somewhere. The B cells, the B1 cells, so there's a big shift here, and it's sort of captured here from the CD4 T cells, CD4 CD8. The B1 cells are almost completely gone. There's neutrophilia that's induced, and so what we're seeing here, that it's also accompanied by this in vivo congruence of realm alpha being this sort of IL-4 stimulated cytokine. RSM and choline inhibition is decreasing the RSM in circulation only after 5 injections of this drug. And in the peritoneal cavity, we're also seeing a decrease in the amount of realm alpha. And just to bring it home in terms of the intestinal burden, which is what this in vivo model affects, looking here at CD206, and looking at the lumen of the intestine, there's really no realm alpha. There's nothing in any of the naive samples. And if we, this is called a primary infection in the vehicle, you can see that realm alpha is expressed, CD206 is expressed here. But if we treat with our RSM, now with the infection model, it's devoid of realm alpha staining and potentially deficient of CD206 as well. So it's not just a localized effect. We're able to sort of modulate this reaction or this relationship almost systemically. So again, how does choline metabolism affect IL-4 responses? Well, it really does just sort of skew Th1 versus Th2 responses. And I'd be happy to talk about it after. But I only want to leave with a quick, I know I'm over time a little bit, but question everything. To all the trainees out there, we were told that that choline was only oxidized in the mitochondria of liver and kidney. And yet there are papers out there showing that it's in other cell types, and that choline transporters, when they are in the mitochondria, have an effect. So right now we're looking at this, and Sierra has shown that choline transporters are actually localized to the mitochondria of macrophages. And so we're now characterizing why that might be and how it might play a role in various different contexts. And through that, we are looking at different patients who have homozygous mutations of CTL1, and they are showing that above all that could go wrong with choline metabolism, it actually is a new type of child onset neurodegeneration that these patients are looking at. So we have these fibroblasts, and we're de-differentiating these as we speak to look at the phenotype. So huge thanks to everybody who's involved, and I'd be happy to take questions, and I apologize for monologuing and going over time.
Great, thanks very much. Great presentation. You know, a lot of work, but that shows that this is why we call the topic research, right? You know, you actually take earlier observations and being able to really bring them into new light, and hence a lot of the comments you made for the trainees as well. And I absolutely agree that you needed to really take everything in perspective, and never take things, especially in papers or textbooks, for granted. Now, let me see if there are any questions. Maybe I can, so in view of time, maybe I just ask a couple questions, but invite people to, for sure, pose your questions, Q&A, or you can, raise your hand as well. So one thing I was interested in the significant changes, for example, in terms of macrophage expression and function, choline uptake, when you actually, change the, even though the overall kind of choline uptake rate is similar, but their fate is quite different. Is there complementary or counter-regulatory processes? Does the fatty acid uptake change in that type of setting? So does does the synthetic pathway, because there are these potential alternatives, change as you change choline availability?
Yeah, and that's an interesting question, because it's something that we sort of only know half of. That's the one side of the equation, and we know that in certain situations we can macrophages upregulate the synthesis of those fatty acids, presumably to incorporate into phospholipids and other lipids and grow. But we also know that under certain conditions, like IL-4 and other situations, they upregulate the transporters that bring in exogenous fatty acids. So I would like to think from an energetic perspective, it's easier to bring in fatty acids and just use them after breaking them down, as opposed to synthesizing them. It's quite energetically demanding to make new lipids, and potentially that's part of what the metabolism rewiring is for, is to be able to generate the energy to make the lipids to be the cell that will sort of phagocytose. So it is all connected, and I think it would be interesting to know sort of the extent to which both of those programs are used in various contexts, especially over time with different conditions. Yeah,
because it may alter the overall balance of the energetics, but also in terms of where those things coordinated, and the targets, for example, you mentioned mitochondria, there may be other organelles, in fact, that may represent shift. And then the other thing I was, now here is really digging back to some ancient things that I probably totally remember wrong. And I seem to think that, the phosphatidylcholine and, these substrates affect the membrane, fragility and rigidity. Is that still true, or am I just totally barking up the wrong tree?
You're not barking up the wrong tree. I mean, it's a big tree and it's there's a lot of branches, but the PC, that phospholipid in particular, I think is more likely to have saturated fatty acids, which will be more rigid than the polyunsaturated. And so the rigidity there, I think, is certainly key. I don't know. I think the sphingomyelins and the cholesterol are more focusing on the lipid rafts, but I think when it comes to rigidity of membranes, for sure, all of the different fatty acids are going to play a role, and the head group actually does a lot to modulate which fatty acids will be incorporated. So the PE is actually much more likely to have the big polyunsaturated branches of fatty acids that will be a bit more fluid as opposed to the PE. So, and it doesn't help the complication of the matter, because then it comes down to, now are we changing membrane fluidity, and that's what's affecting transporter function.
So, because it's always fascinating in terms of the proteins that's existing, in the membranes, versus the overall biological function. So now I'm going to just maybe ask a final question, that is the translational aspect. So there's a bit of a paradox, in choline. I kind of keep an eye on it from the side, because it's a fascinating aspect, in terms of immune response. So certainly it looks like if you have a deficiency, you just showed the example at the end, so you can get neurological complications. There's suggestion that you can get cognitive impairment if there isn't adequate intake. Certainly you can get liver dysfunction, as you mentioned. On the cardiovascular side, it's a little bit of a split, so it's a little complicated depending on which camp you're in, whether you think the TMAO, is important or not, and things like that. And so at the end of the day, are we getting enough choline in our diet? Knowing that eggs and meat are getting pricier all the time. And so what should one do to maintain good health from choline point of view?
Well, with the asterisk being, and the disclaimer that I am not a physician, I am not putting this out there as a health benefit. There are a lot of studies that contradict each other when it comes to choline. Choline can exist in its free form, it can exist in phospholipids, and we're ingesting both of them. And it's been shown, Renee Jacobs' group from Alberta, they've done a study in atherosclerosis where they supplemented mice with phosphatidylcholine or with just free choline, and it was only the phosphatidylcholine mice that showed a protection and had changes in various things that were beneficial. And so I think that it's hard to know whether or not everybody is deficient for choline, and by increasing any form of it, we would be protected. So it could come down to some sort of a personalized thing as well, because I know that the various intakes differ based on individual need as well. So it does not help uncomplicate things, but it certainly keeps it interesting.
I think you mentioned like pregnant women, there are situations, right, where the relative choline deficiency, exists, and that's particularly the setting where, adequate choline intake will be important. Wow, okay, very exciting. So we are constantly learning, and and I think this particularly shows the importance of the interdisciplinary type of research, right? I really admire the fact that you have a problem and you just go and find out who the experts are around, and just go and get the best tools from them and solve.
Some might call it lazy, but it's been
it's actually fantastic, because you help them to solve their problems too, right? So this is really actually how you innovate. yeah, it's been this is how innovation takes place. I love the story on the dengue fever. I just love it. Yeah, so so thank you so much, and all the best to the ongoing work, because this is obviously an area that is important in health, and very important in terms of sort of practical implications on the immune side, on the metabolic side, and on the cell function side. And so it's really very exciting, and I hope that you will be able to come back in a year to tell me exactly what we should eat, so that we really enrich our anti-inflammatory repertoire. Okay. So yeah, so thanks so much, and I really appreciate it, and all the best to your trainees as well, and thank you for spending time with us.
Thanks very much.